Thanks for joining me for another edition of Parker Biology. Today we're going to do a quick overview of photosynthesis. This is not intended to replace our notes from class, just to help us review some big ideas. The first big idea is, where are we when we talk about photosynthesis? Well, we're in the green parts of plants. So specifically here we're looking at some leaf tissue. And this middle region of the leaf can be referred to as mesophyll cells. We can see some vascular tissue here delivering materials, and also the really important openings, typically on the underside of a leaf. These are called stomata, and these allow for gas exchange. So CO2 can enter and oxygen gas can leave. As we dive further into some of these mesophyll cells, we can see that there are little organelles specialized for carrying out photosynthesis, and these are called the chloroplasts. Now within each chloroplast are a series of thylakoids, small coin-shaped disc-like structures that form stacks called grana and are surrounded by a thick liquid material, the soupy part of the chloroplast, if you will, and that's called the stroma. As we discuss the light reactions in the Kelvin cycle, we'll see that those occur in the thylakoids and stroma, respectively. When we think about the overall reaction of photosynthesis, we know that plants are breathing in CO2, taking in water, and ultimately we hope that they make their own sugar, or glucose. We also know that oxygen is produced as a byproduct. So we looked at the Van Niel experiment in class, and we realized that this oxygen that's released in photosynthesis is actually coming from water molecules. In a process known as photolysis, using light energy, water molecules are split, the hydrogens will be further split, and the oxygen molecules will be released as a byproduct. Another really important piece of background information has to do with redox reactions. Electrons are going to be excited and bouncing down electron transport chains through photosynthesis, and as they do so, as they're transferred, some substances will be oxidized as they lose electrons, and some substances will be reduced as they gain electrons. Now, what's nice for us in biology is that electrons don't always move alone. If an electron is traveling with a proton, well, that's a hydrogen atom. So instead of just looking for electrons or E's with a negative sign, we can also follow what happens with H's. So specifically, as I look at the photosynthesis equation, and I see that carbon dioxide is gaining H's, oil rig reduction is gain, we can say that carbon dioxide has been reduced. Also, if I look at water and see that it has lost H's, oil rig, I can say that the water has been oxidized because it has lost those electrons or those H's. Our goal in studying photosynthesis is to better understand how the reactants become converted into these products, how water is going to come in, resulting in oxygen being released, and how carbon dioxide is going to get built into sugars. We will be breaking this into the light reactions, whereby solar energy is converted into two forms of chemical energy, ATP and NADPH. And then we'll be looking at the Kelvin cycle out here in the stroma, where CO2 will be built into sugar by spending the energy provided by the light reactions. Notice that Kelvin cycle will return the lower energy forms, ADP, inorganic phosphate, NADP+, back to the light reactions to get re-energized. We'll be seeing how interdependent these two types of reactions are, the light reactions in the thylakoids and the Kelvin cycle in the stroma. The last piece of real background information to think about is the idea of pigments. Pigments are substances that absorb and reflect light, typically different wavelengths. And that's going to be important because we're looking at solar energy being absorbed. So an absorption spectrum allows us to plot how good a pigment is at absorbing different wavelengths. So here we can see chlorophyll A's peak absorption, and then we notice it really drops off. We see chlorophyll B's peak, the carotenoids peaks, but notice that there doesn't seem to be much absorption at all in this region. And the secret to that is, if a pigment is not able to absorb, it reflects. And that helps explain why plants appear green, because these green and yellow wavelengths are reflected by chlorophyll, not absorbed. What's nice is that by having multiple pigments, it's going to make the plant a lot better at absorbing all of these wavelengths instead of just a narrow region here or a narrow region here. Much more efficient, more wavelengths absorbed. As nice as it is to see which pigments are absorbing which wavelengths, it helps us more to see how effectively each wavelength is at driving a biological process. And that's reflected in a graph called an action spectrum. This shows us that there is a lot of oxygen being released, and therefore a lot of photosynthesis happening at these wavelengths because they're being absorbed. Not much photosynthesis happening here, and that correlates to the idea that these wavelengths were reflected. And then we start to see another peak where photosynthesis is occurring well again, and that again correlates back to this idea that if that energy is absorbed, then the organism can do something with it, like carry out photosynthesis. While modern action spectrums use quite a bit of technology, Engelmann was really resourceful back in the 1800s. He just used a piece of algae. And he put the piece of algae with a bunch of different wavelengths of light separated out by a prism, and he grew that algae in the presence of aerobic bacteria. Now, aerobic implies that these bacteria required oxygen, and we can see that in areas where they grew especially well, oxygen must have been plentiful. What process would have provided that oxygen? That's right, photosynthesis by the algae. So as the algae cell was able to absorb certain wavelengths, it was able to carry out photosynthesis, releasing oxygen and therefore allowing all these bacteria to grow. Notice this region, though. It doesn't appear that aerobic bacteria were able to grow. That implies less oxygen because of less photosynthesis. And why does that make sense in the green region? Well, green wavelengths are reflected by these algae. Since those wavelengths were not absorbed, photosynthesis was not happening here, oxygen was not released, therefore those aerobic bacteria were not able to grow. Pretty amazing little setup, and if you notice the curvature coincides with that modern action spectrum. Again, seeing how well different wavelengths are able to drive the process of photosynthesis. Now we're going to dive further into the light reactions. Remember, big picture, light reactions should be converting solar energy into chemical energy. Here we can see the light energy coming in, photons, causing a transfer of energy. There's some resonance going on here within these light harvesting complexes. And at the reaction center complex of the photosystem, electrons will get excited and jump to a primary electron acceptor. Notice we are embedded in the thylakoid membrane here, and this is all a protein complex. There are two main types of photosystems, Photosystem 2 and Photosystem 1. Photosystem 2 is lovingly nicknamed P680. 680 nanometers is its optimal wavelength, whereas Photosystem 1 is nicknamed P700. Its optimal wavelength for absorption is 700 nanometers. This diagram is depicting the linear electron flow. It's going to bring about production of equal amounts of ATP and NADPH, again converting that solar energy into the two forms of chemical energy. As the photons come in, resonance energy, electrons getting excited. Here we can see we are at photosystem 2, or P680. So these P680 chlorophyll molecules end up getting their electrons really excited by the solar energy. So excited that they jump to a higher energy level. And that leaves a void. That leaves a hole that needs to be filled. And that hole is going to be filled by the splitting of water molecules. The H's will split. Protons will end up in the thylakoid lumen, or space. And the electrons from the water are going to be used to fill that hole created where those excited electrons left. What's left over? If the H's are involved here, the oxygens are going to be given off as waste. And we knew that. We knew that the sun's energy was important, that water came in, and oxygen was released as a result of the splitting of water. Now what about these excited electrons? They got really excited and jumped up to a higher energy level, and from here they're going to bounce down a series of complexes embedded in the thylakoid membrane, and we call this the electron transport chain. We'll look at the mechanism in a minute of chemiosmosis, but ultimately the bouncing of those electrons down the ETC will result in the production of ATP. Now as they reach the end of their chain, those initial excited electrons are filling in another void, another hole, left in photosystem 1. A different photon excited electrons, leaving a hole that is filled in by the falling electrons from photosystem 2. We have a second electron transport chain that results in production of NADPH. So yes, equal amounts of these two forms of chemical energy are formed through these linear electron flow pathways. Because Kelvin cycle requires a little extra ATP as compared to NADPH, sometimes the cell needs to switch over, and instead of carrying out this linear electron flow, it needs to utilize what we call cyclic electron flow. This is only involving photosystem 1. Excited electrons jump up, and instead of getting passed down this shorter electron transport chain, this is where we said in class, psych, it passes back down the chain, and that extra ATP will be formed here. Again, that's due to Kelvin's greediness. He needs a little bit extra of that form. Here we have a diagram that shows us the same story, the excitement of electrons, the bouncing of electrons, light energy to chemical energy. But it gives us a little more of a hint at how that electron transport chain actually drives ATP synthesis. Every time the electrons bounce down the chain, that bouncing is exergonic. A little bit of energy is released with each bounce. And that allows for the pumping, the active transport of protons. As protons build up inside the thylakoid, we call this the thylakoid space or the thylakoid lumen. And as protons build up, their natural tendency is to try to move from an area of high concentration to an area of low concentration. Because they're charged, they can't just squeeze through that thylakoid membrane. The only place that these protons can actually shoot through is a very specialized enzyme called ATP synthase. This is a rotor, a revolving door, and as the protons fly through, this whole unit spins, uniting inorganic phosphate with ADP, thus forming ATP. What's great here is it shows us that these chemical energy forms are actually now out in the stroma, which is perfect for them to make their way over to the Kelvin cycle. I'm going to end the video here with a quick summary of the light reactions, and our next will help summarize the Kelvin cycle and some special modifications that some plants have.